Thank you, Diana, for introducing me. Hello, everybody. It's good to be here. It's my second time here, and I see some old friends, and I hope to make some new friends too. So, yes, my name is Maxim Freydin. I'm a lecturer in Genetic Epidemiology in King's College London. Here we have my web page. If you want to know about my research, my publications and all that, you can go there. And of course, you can ask me, we can chat about everything. And also my email, if you have any questions, feel free to drop me an email. And of course, feel free to approach me while I'm here. So I will tell you about genetic epidemiology and focusing specifically on genome-wide association studies. Before doing that, because I didn't have a chance to introduce myself on one of these, what is it called, drink and chat session. Right, so I put this slide here. So I'm originally from the city called Tomsk, which is in Russia, in Siberia. I hold PhD in genetics. My current position, lecturer in genetic epidemiology at King's. My research interest is genetics of complex human diseases. So diseases which are driven by multiple genetic factors, which interact with multiple environmental factors. So this is what we call complex human diseases. I used to work with various phenotypes of this kind, including allergy, infectious diseases, cancer, radiosensitivity, pain syndromes, and pain-related phenotypes, and some anthropometric factors. So my current research projects are focused around genetics of comorbidity and inverse comorbidity. So comorbidity diseases and phenotypes, they co-occur, clinical phenotypes, they tend to co-occur, they come together in a single patient. Inverse comorbidity is a very interesting situation when diseases inversely correlate. If you have one disease, you will never have another disease if they are in inverse comorbidity situation. And I'm interested in genetic background of this phenomenon. My current project is the genetics of lifetime disease burden. So when we age, we accumulate diseases, we accumulate diagnoses, and actually how many diseases will we have depends in many, to a large extent, depends on our genetics. And I'm looking for genetic factors which driving the number of diagnoses which we have during our life. So I also do some teaching duties in medical genetics and statistics and run some short courses on genetic epidemiology. And also I am a specialty chief editor for Frontiers in Genetics journal for the section Human and Medical Genomics. So if you have a paper in mind, so feel free to submit and I will give you some, preferences. As long as your paper is good. Anyway, so that's my introduction, and let's talk about the plan for today. So I will give you introduction into the wonderful world of genome-wide association studies. Then I will tell you briefly about some useful applications of genome-wide association studies beyond identifying genetic factors underlying diseases, in particular polygenic risk scores and the Mendelian randomization. And we will have a workshop when I will show you how to run simple GWAS models. During my lecture, during everything, so feel free to interrupt me, ask me questions. I'm more than happy to run it interactively rather than just me constantly talking. Okay, so let's start from introduction to GWAS, but first, a few words about genetic epidemiology. It's a branch of science that studies genetic factors underlying health and disease in families and populations. It's been born as an interplay between epidemiology and population genetics. In 1950s, it seeks answers to three major questions. Is there a genetic component to the disease? If so, what is its relative impact of genetic component? And what's the actual genetic factors? To answer this last question, there are two major approaches, linkage analysis and association analysis. So linkage analysis I'm not going to talk about. It's based on the observations and some calculations of co-segregation of genetic markers and disease in families. It was particularly useful for monogenic disorders, not very useful for complex diseases, so that's why I'm not going to talk about that. And I will talk about association analysis. So the central concept to answer first two questions, how we do that in genetic epidemiology, we estimate what we call heritability. Does anybody know what this is? Yes? Possibly you heard about it. Heritability is the proportion of the variance of a trait which is attributable to genetic factors. So this is the distribution of height. You see the mean value and some variation around it. To some extent, this variation around mean is because of a difference in the population in genetic factors, and to some extent because of the difference in environmental factors. So we can decompose this variation of the phenotypic trait into these two components, genetic variance and environmental variance. And we can calculate two types of heritability: broad sense heritability, usually designated by capital H, which is the proportion of total genetic variance to total phenotypic variance. And narrow sense heritability, which is usually more interesting thing, is a proportion of additive genetic variance. I will tell you a bit more, some details about that a bit later. But just in brief, so when we have a genetic factor slightly contributed towards a phenotype, then we have another one and another one. Their effects can be summed up, and this in additive manner. This would be additive genetic variance. And another part is dominant genetic effects, when we have, let's say, single mutation, strong mutation with a very strong effect, which doesn't sum up with other factors, which actually makes you having this phenotype. So genetic variance can be decomposed into additive and dominant variance, and narrow sense heritability is additive genetic variance divided by total phenotypic variance. Apparently, the higher heritability, the more evidence for this trait to be driven to a large extent by genetic factors. So that's why we actually estimate it. So how can we do that? The classical way is to look at similarity and dissimilarity between monozygotic and dizygotic twins. So monozygotic twins, as you know, they share 100% of their germline genetic variation, so they're genetically the same. And the dizygotic twins, they share about 50% of germline genetic variation. So when you say 50% is just average estimate, in fact, it can be anything from 0 to 100. And actual estimates from sequencing about is the range is between 33% to 60%. This is what dizygotic twins share between them. On average, smaller than monozygotic twins. So, and when we compare similarity and dissimilarity in monozygotic twins and dizygotic twins, we actually can calculate heritability in a very simple manner, like that, double the difference of correlations between monozygotic twins and dizygotic twins. It can be correlation if we talk about a quantitative trait, continuous trait. It can be what we call a concordance rate. So concordance means similarity between twins for discrete traits and diseases. So this is a very simple formula, and nowadays we don't use it. But it's just to illustrate how can this be calculated. So if concordance or correlation of monozygotic twins is higher than for dizygotic twins, we assume that disease has a genetic component. If the concordance or correlation are not so different, so disease is determined largely by the environment. For cases when correlation between monozygotic twins is high, 100%, disease is actually determined by genetics. Not even there is a genetic component, it's determined. Okay, this is what drives variation of this trait. And if for monozygotic twins correlation less than 100%, disease also has an environmental component. This is how we kind of treat twins and how we can make assumptions about genetic component of or impact of genetic or environmental component into the disease or a trait. Oops, that's the wrong one. So as I said, we don't normally use that simple formula anymore. We use a much more complicated model, which is called ACE model. A stands for additive genetic factors, C for common environmental factors, and E for random environmental factors. So common factors, factors which people share when they live in the same house, eat the same food and all that. random environment, it's something may happen to a particular person, not necessarily to the other one. Okay, you go out and you break your leg. Okay, this is a random environmental kind of factor. So, and we assume that there are some latent factors, additive genetic, common environment, and the random environmental factors. To some extent, genetic factors would correlate for monozygotic twins and less correlate for dizygotic twins. We assume that shared environment is the same, and then we use an approach which is called structural equation modeling. It's a form of factor analysis to decompose phenotypic variance into these latent factors, components of variation, phenotypic variation. The beauty, it's technically now simple. It used to be very complicated. Now it's very simple. There are R packages which can do it very easily. What's the beauty of it? Unlike this equation which I showed you before, it allows adjusting for all sorts of covariates. Any questions so far? No questions, okay. So some examples of heritability. Blood pressure, the correlation for monozygotic. It's not for every single population. It's an example for some estimates for some population. Blood pressure 0.58 for monozygotic twins, 0.27 for dizygotic twins. Correlation between monozygotic is higher. Heritability would be around 62%, which means that, yes, there is a lot of genetic variation attributable to this trait. If you look at things like height, the heritability will be very high, almost 100%. In many cases, like up to 90%, almost up to 100% in many cases, which means that your height is to pretty much, to a very high extent, is determined by genetic factors. So whatever you do in your life, your height is kind of driven by genetic factors. Yes?
Could you just tell how concordance rates are calculated?
Concordance rates, how they are calculated?
Yeah, it's just
this is a correlation. This is simple intra-class correlation. Concordance is the similarity. You have a pairs of twins, many pairs of twins. You have twin pair number one. You see if they have the same phenotype, they're concordant. Then you calculate how many pairs with the same phenotype you have, right? And divide to the total number of pairs. This would be your concordance. Okay? It's very simple. Yes?
How does it work with measles?
In measles, yes. I was coming to it. They do correlate both monozygotic and dizygotic twins. Measles infectious disease. It's very likely if I have this infectious disease, my twin brother or sister would also have it, because, right? But in any infectious disorders, there is also genetic component. Not everybody who is exposed to a virus or a bacteria would get the disease. Okay? So yes, there is a subtle genetic component here, but it is largely more, the impact of environmental factors is much bigger here.
No, I mean, how are concordance rates estimated?
Concordance, well, it's
You said it's correlation with how, what kind of...
concordance rate. We have pairs of twins. One twin has measles, another has measles. They are concordant. Another pair, one twin has measles, but the other one does not. They are discordant.
So it's fraction of...
Factors of the fraction of those who share the disease. Yeah. Yes?
What is can be right? So we need to pretty large sample of twins?
Yes. Yes. We do need a large sample of twins, the bigger the better, and measurements of these phenotypes in all twins in each pair.
I mean, it's a common task to find such sample.
It's a very simple task nowadays. You can approach me. I work in the department which runs the resource which is called Twins UK. We have a collection of twins, 50,000 twins, it's about 9,000 twin pairs, heavily phenotyped, genotyped, with lots of omics. There are other resources like that, like Swedish Twin Registry, for example. There are many twin registries in the world which actually you can approach, get the data, do some analysis. Yeah, apparently you can actually start collect. In fact, in Poland, they started recently a Polish twin registry. So soon it will be available here.
how difficult it is to get a general picture regardless of the populations investigated. For example, I see that there is the example with height, where the heritability is around 1,
Yep.
but I assume that this could be true for quite a lot of countries where nutrition in childhood is almost at the sufficient level
Yep.
for most of the people. So is it the same for countries where this is really a problem, socioeconomic status,
Yep.
and so on? And how can we interpret the heritability value that way?
So two things here. First, heritability estimates are specific for the particular population we work with, because of environmental factors can be different in different populations, as you mentioned. Genetic factors also can be different in different populations, so relative impact may vary. For many traits, the heritability estimates will vary accordingly. For traits which have been heavy subject of natural selection, like height, the heritability estimates are more or less similar. They may be slightly lower, slightly higher, but more or less similar. For traits which have not been subject of natural selection, which allows actually much bigger variation, weight, for example, right? So weight there's a strong genetic component, but it really depends on how much you eat, okay? So in your example, if we take different countries, the thing is that we look at people living in the same environment. Okay, they're exposed to the same factors, they live in the same family and all that. So the impact of these differences between population may not necessarily be very strong. It may be, maybe not. So yes, that's why we need to calculate heritability in every single population we're interested in and see what happens. And then we follow our subsequent analysis based on that. Any more questions? Okay, let's move on. Right, so okay, let's say we found a trait which is highly heritable. So we answered first two questions. So yes, there is a genetic component and there is a reasonably high impact of genetic factors. The next step would be to determine these genetic factors, right? And I mentioned leakage analysis. We don't consider here. We look at genetic association studies. Genetic association studies, a very simple thing. It's actually the test for relationship between a disease status and genetic variation. The aim of genetic association studies is to identify genes or some genome regions which contribute to a specific disease. And this is done statistically in a very simple manner at the background as a comparison of the prevalence of allele frequencies in groups of people, which we call cases or controls. So cases are people who have the disease in question. Controls are people who don't have the disease in question. So in this example, we have 1,000 cases, people with some kind of heart disease, and we have 1,000 controls, people without this heart disease, and we have a single SNP, single genetic variant, C allele or T allele, and we see that C allele is much more common in cases compared to control, 62% versus 49%. If it's all statistically significant, we can say that allele C is associated with this disease. We can also reverse it and say allele T is protective against this disease. All right? So this is a very simple illustration of the principle of association studies. If we look at the continuous phenotype, so we can see that adding one copy of an allele would increase slightly the level of a continuous trait in those people who have it. And if we have two copies of this allele, this goes even higher. So this is illustration of additive effect of alleles. So those which are AA would have this level, AT on average higher level, TT even higher level. Okay, this is very kind of simple thing in the nutshell. In practice, of course, it's much more complicated, but this is how it works. So how do we choose what to analyze, what genes, what genetic regions? First approach is candidate gene studies, which is driven by a hypothesis. Let's say we already know something about pathophysiology of this disease, and we're interested in studying particular genes which we know are involved into realization of this phenotypic trait. So in this case, it's polyaromatic hydrocarbons, which are known to be associated with the cancer. And we know how they are metabolized. And we know what molecules, proteins, factors are involved. And we may suggest, so let's say the genetic variation in the levels of this protein might be related how this protein active or how much protein is released, and depending on that, we expect some phenotypic manifestation. So we take genetic variation in this particular gene, selected based on a hypothesis, and test for association between genetic variation in this gene and the phenotype of interest. This is candidate gene approach. It can be powerful, but apparently we really need to know, or at least have some strong hypothesis about the mechanisms of phenotypic manifestation of a trait, which is apparently not always the case. We can do it the other way, using genome-wide association study, which is a data-driven approach. We don't have any hypothesis about phenotypic mechanisms of this phenotype. We just simply scan the entire genome for the signals of association. And when we find the signal, we look what genes in there. And then we generate a hypothesis that this phenotype is associated with this gene or this gene or this gene. So this is genome-wide association study. This is the trend of kind of the use of genome-wide association studies in the literature. It comes to 2015. I didn't find the graph to show 2025, but it should be somewhere possibly here. We have hundreds and hundreds of genome-wide association studies being released. We have a consortia which keep producing genome-wide association studies. So this is our main instrument nowadays because it is very powerful, plus we have large samples available, and it produces good results. Okay, so we look for association with phenotype without any hypothesis. We test multiple genetic factors across the entire genome. Usually these are single nucleotide polymorphisms. And this approach helped to identify like thousands and thousands of genes for thousands diseases and phenotypes for the last 20-25 years. So very powerful approach. So how this is done? We start from the data collection. We may already have the data collected. For example, there are biobanks which already collected all the data and even genotyped for you, the participants. But let's say we just started things from scratch. We do a data collection. We find some collaborators in the hospital who collects for us data, or run some big population study, collect thousands of individuals, with or without the phenotype of interest, depending on the situation. Then we carry out genotyping. Then we do some quality control to identify genetic ancestry of our population. I will explain you why this is important. Then we do what is called imputation. We cannot technically genotype 20 million SNPs. Right? It's technically very challenging, but we don't need to. What we need to is to genotype, let's say, 1 or 2 million of so-called tag SNPs, which we use to restore SNPs around them based on the linkage disequilibrium. I will tell you again details. Then we carry out association testing, producing graphs like that Manhattan plot, which shows signals for association. Then, if possible, we do meta-analysis and replication analysis in some separate sample. And then we do most interesting bit, post-GWAS analysis, because what we end up with is actually a list of SNPs with some metrics of statistical significance and effect sizes. Okay, now from these SNPs, we need to identify genes they are related to. And then when we identify the genes, we need to identify pathways and networks these genes are involved to. And then we can explain the phenotypic manifestation of this disease. So this is actually most important and most interesting part of GWAS. Let's talk. Yes?
Speaking of imputation, so if you impute SNPs, it means that they are not independent.
Yes. I have a few slides about that to explain. Yes, but SNPs, if they are in linkage disequilibrium, they correlate with each other. So if we know one, we can make a very good guess what would be the one next to it.
Sure, but that complicates the interpretation of key values and hypothesis testing in general.
It does not complicate.
Because there's dependency of hypothesis, not independent.
Yeah, exactly. Again, so I will tell you about that as well. Yes, yeah, I know what you're talking about. Yes, there is a solution for that. Yeah.
Can you comment on batch effects in GWAS and how to deal with them?
Batch effect.
Yeah, because there's such a big sample size. So
Yes. So first, it depends what you mean by batch effect. But yes, they can be modeled in your regression model. We put the batch effects in the regression model if we found these effects and adjust for it. Okay. So data collection in most of the GWAS is it's unrelated individuals. So now, in most of the time, we can also use families, and when GWAS started, people used families, but they're difficult to collect compared to like a population. And so we used them before when they switched to unrelated individuals, but actually now we're coming back to families because there are very interesting approach to GWAS, which specifically tailored to families. So it's a kind of new trend switching back to families, but as I said, they're difficult to collect. So genotyping, so just examples of microarrays. This is an outdated version of Affymetrix microarray. Now they have like plates with many arrays on a single plate, and this is Illumina. We have 12 arrays on a single plate, so we can genotype 12 individuals. But so typically we use Illumina or Affymetrix microarrays. They have roughly 1 or maybe 2 million so-called tag SNPs. Also, apart from the genotyping, we can use results of whole genome sequencing and whole exome sequencing, so we don't need to use any imputation. We just simply identify genetic variants directly. It's still rare because it's, in mass of big GWAS with hundreds of thousands of individuals. It's very expensive. But the solution now is to use low-level whole exome sequencing followed by imputation. So this is a new trend, most recent and very promising. It's not so expensive and provides a kind of very good solution for that. So ancestry analysis, quality control. For some statistical reasons, our sample must be genetically homogeneous. Otherwise, we may end up, reasonably. Otherwise, we may end up what is called a population stratification. And these end up with false discoveries. So what we need to do to avoid that is to project our sample on a principal component graph along with some reference population, which genetic ancestry we know. There are several projects which actually provide you this data, like 1,000 genomes that have data for many populations from Africa, Asia, Europe. And let's say this is our reference European sample. So this green is our sample. So this is where this is the genetic ancestry of our sample. So these are South Asians, so Indians, Pakistanis. So these are Africans. Right, and these are East Asians. Oh, sorry, no, this is East Asian, Southeast Asian. This is South Asians, Indians, Pakistani. This is Chinese, Japanese individuals. So if our sample here, let's say we are interested in Europeans, so we have these people here, which seems to be like Africans or Asians. We better exclude them from our analysis. Okay, as a starting point. We can go deeper, we can analyze, let's say we have Europeans, we can split them into Northern Europeans, Middle Europe, Southern Europe, depending on our interests. Okay, so this is ancestry analysis which we do. So, imputation. based on the phenomenon of linkage disequilibrium and the presence of haplotypic blocks, haplotypes. Okay, so this is the example here. We have like 6 SNPs, A or G, C or T, A or C, A or G, A or G, C or G, and T and A. If there is no any correlation between them, every combination between these SNPs is equally possible. But here we have this combination A, C, A, G, T, 35%, which is much more than we would expect if they have been randomly allocated on the genome, 30%, and we have several other haplotypes. So they are correlated. Okay, they come together as a single unit, which is called haplotype. So haplotype is a group of alleles on a single chromosome, which are inherited together. And if we look at the genome, which if we calculate correlation between the SNP, we will see these blocks of correlated SNPs, LD blocks. Okay? Important thing, these LD blocks may not necessarily be the same in different populations. If you look at Europeans and if you look at Africans, many haplotypic blocks are strikingly different. Okay? So, if we know LD structure, if we know the haplotypic structure, we don't need to genotype all these SNPs. We need to genotype only one here, and then we predict SNPs based on LD pattern, and we restore a haplotypic block. Okay, so coming to a question, because they are correlated, we do not adjust for every single number of tests for the total number of SNPs which we restore, genotype and restore. We adjust for the number of haplotypic blocks. So this would be the number of independent tests in GWAS. Okay? So,
sorry, may I ask a little question? Say the borders between the blocks are the crossover hotspots, or?
It's crossing, not necessarily hotspots, but, well, yes, these are spots of crossing over, yeah. Yeah. Okay, so, of course, we need to know reference haplotypes in populations, and there are several big projects which actually have this aim to identify, describe the patterns of linkage disequilibrium and haplotypic blocks in various populations worldwide. So just to mention a few, the International HapMap project was the historically first one, finished in 2010. 1,000 genomes, finished in 2015. Haplotype Reference Consortium is still ongoing. The Genome Asia, 100,000 K, started several years ago. It's a very important haplotypic consortium looking at Asian region in particular and different Asian populations. And there is also the African one, or several African ones, also tailored to African populations. So what they do, they generate reference maps of haplotypes and list of SNPs in LD with LD metrics across different populations, which is important for analysis of genetic ancestry, for historical things, for analysis of evolution of human populations, and of course for gene mapping and association analysis.
I have a question about that.
Yes?
Is there a suspected reason why haplotypes would differ? Not that they would differ, but their size or the correlations themselves would differ between populations?
Oh, yes. So the population, they spread around the world from different, they live in different places, they have different genetic and environmental factors. Mutation, migration, natural selection, genetic drift, all of these things, they shape the genetic variation, haplotypic structure in different populations.
What I was more referring to is that aren't those haplotypes, at least the size of the blocks, defined by some biology?
It defines by evolutionary population genetic factors and molecular factors as well. The recombination kind of places where recombination tend to occur in human genomes.
Yeah. And this can also differ between populations, even those.
Okay, so it's reasonably similar. Let's say if we look at Europeans, it's more or less similar within Europeans. compared to Africans, for example, or Asians, and so on. Then, and then, as I mentioned, so if we can narrow this down, we look at Northern Europeans, Southern Europeans, and all that. So factors which I mentioned, evolutionary factors, factors of population dynamics, mutation, migration, drift, nature selection, all that. They all shape the patterns of linkage disequilibrium in different populations. And history of population. If this population, we have this population living here, we know it originated from that one. We would expect a high genetic correlation between them, high similarity. Yeah. Then the further the more distant they are from the ancestry population, the more genetic variation is reshaped, accumulated, changed, and so on. So if we assume we come from Africa, so originally people who stayed in Africa, they have one pattern, and then after like thousands of years of evolution, other populations, they changed dramatically in terms of genetic variation.
So this is not that we can generalize some of those findings across all of them.
No, we can. Yeah, well, that's a very important thing. So we cannot simply generalize. Okay, so that's why we have to do this analysis in different populations and see what is population specific, what is actually general.
Okay, I get that.
Yep. All right. So reference panels, which we can use for imputation. Some software, just to mention, Impute4 and Beagle and Minimax. So it's statistical, technical. It's very it's challenging procedure because you have to do lots and lots of computations. You cannot do it on your laptop. You have to use imputation service. And just to mention a few, Michigan imputation service server, Sangha, TopMed, because it's very time and resource consuming. Just to give you an idea, we have a group of people which we genotype for a limited number of SNPs, our tag SNPs, okay? And then we have reference haplotype, and we want to know what are the genotypes here, in whether there are question marks. So we can use this reference haplotype to restore haplotype in these people here, okay? So I don't know, I think it's pretty small, but this is our study sample. We only genotype 3 SNPs here. Okay, then these are reference haplotypes. This is what we can expect in this population with this frequency or that frequency. Okay, and actually we make this analysis looking, if we have A on this chromosome, it's very likely that this haplotype will be, the haplotype here for this person will be this one. Okay, because we have these genotypes here. Here, slightly more complicated, we have GCA. G and C correspond to this haplotype, and A corresponds to this haplotype. Okay, so based on this genotypic information and reference haplotypic structure, we restore the haplotypes and genotypes in this study sample. Right.
I have one more question, sorry for interrupting you, but okay, so we have those sequences like populationally conserved in a way that we can, knowing some of them, deduce the rest of it. But wouldn't that be like an amazing place for some very painful mutations to happen in because it is conserved? So when it's broken, then it's not...
No, of course, absolutely. Mutations do happen, so we can have, but they are rare. Okay, and what are the chances for mutation? We analyze it at 100,000 people. What are the chances that this mutation happen in like a large amount of people? It's rare. Of course, this can happen. Yes, absolutely. It's a statistical approach which uses available information to try and restore haplotypes. Of course, it cannot account for everything which may happen. Of course, there can be some sort of a weird recombination in this particular person, or I don't know, some structural variant or whatever. Of course, yes, this all may happen. Yes.
So let's say you're designing the GWAS for a specific population.
Well, yeah. So I told you about genetic ancestry, right? We identify genetic ancestry. We say our people are mostly Europeans. Then we use European reference haplotypes to restore. If our people are Africans, or if our people, I don't know, Mexican, or if our people are, I don't know, Chinese or whatever, we use the corresponding reference haplotype based on this genetic ancestry analysis. That's why it's a very important first step to identify who we deal with. Nowadays, there are some sophisticated algorithms which actually allow us to use pretty much everybody in our sample with use of some trans-ethnic meta-analysis approaches, and plus some kind of, smart ways of imputation, even if our population is not entirely like genetically homogeneous. So we come to the point when we can analyze everybody without kind of worry too much about their genetic ancestry, in the analysis. Okay? So what is returned as a result of imputation? Dosages. What we call dosages. What is a dosage? It's a linear transformation of posterior genotype probability. Because it's a statistical algorithm, we cannot say for sure that we have this genotype. We estimate probability for having this genotype in this position. So let's say we have a biallelic locus with two alleles, and A and B, and each individual would have either 0 or 1 or 2 copies of allele B. Let's say we identify genotype probabilities in these individuals like AA 0.1, AB 0.3, and BB 0.6. Now we simply multiply 0, 1, and 2 by 0.1, 0.3, 0.6, and the return is the dosage, 1.5. Which means that in this individual, we have, as we estimated, 1.5 copies of this allele B. Of course, physically it's not possible, right? Of course, we can round this to 2 and say this is a homozygote individual. This would be called a hard call, and in some cases we do that. But from mathematical perspectives, from statistical perspective, using dosages provides you much more power for your genome-wide association study. That's why we use dosages wherever we can, which accounts for the uncertainty of genotyping and imputation. Okay?
Yes, sorry, just to genotype probabilities that you got from imputation.
Yes. Yeah. So these probabilities, this is what imputation server produces, and then and the server also transform it into the dosage. Of course, it's not perfect, and we should have some quality metrics, and they do exist. They're based on different ideas, like proportion of correctly imputed and best guess genotypes to all imputed genotype, or correlation between the dosage and the true genotype. Okay, so, and this just to mention some of these metrics, info and mark 2, they vary from 0 to 1. The bigger, the closer to 1, the better our imputation, of course. And practically, when we run a GWAS, we need to set up some threshold for the dosages we believe in, for imputation quality we believe in, something above 0.5. For example, I tend to use something which is above 0.7. 10, 15 years ago, people tend to use something much smaller because there were not so many SNPs which we could analyze, like 0.3. People consider enough evidence for reasonably good imputation. Any questions about that? Let's move on. And actually, once this is done, we can just run association analysis. Okay? Historically, we use a simple contingency tables, like chi-squared or Fisher's exact test, to look at the proportions of alleles in cases and controls. We don't do this now. We use regression approaches, linear, logistic, Cox regression, depending on the phenotype of interest. We can control for confounders. We can control for population structure, so for some subtle differences in genetic ancestry in our population. And the best one, which we use now, it's linear mixed models. It's also a regression approach, which does the same, but also additionally adjusts our calculations for hidden relatedness. We are all more or less related to each other. bigger extent or less extent. So if we have some relationships in our population which we are not aware of, we can adjust for these relationships via so-called kinship matrix or genetic relatedness matrix. And this is what linear mixed effect models allow us to do. And genetic models we can test different. Normally we use an additive genetic model. This is a standard where genotypes are coding like 0, 1, 2, or like dosages. And they are preferred. And other models are possible: dominant, recessive, overdominant, codominant, and simple comparisons of alleles. They are also possible, but they are not really used apart from some specific cases. So additive genetic model is a standard in GWAS. Statistical significance. This is 5 by 10 and minus 8, the genome-wide significance threshold, which comes from the approximate number of haplotypes in European human genome, about 1 million. So 0.05 divided by 1 million. This is Bonferroni correction for 1 million tests, assuming that we have approximately 1 million haplotypes in a European genome. It does not apply to African genome. In African genome, average African genome, there are about 2 million haplotypes. Okay, more than that. So when we analyze Africans, we have to reduce it by 2 million. Of course, we can use some what we call suggestive threshold. We just don't have enough power, statistical power, to show signal for associations, but people want to publish a paper with their finding. They spend a lot of time and resources, so they say, okay, fine, we didn't achieve statistical significance, but this one is very close to the significance threshold. We call it suggestive. So, and we can, analyze it further. Software, I put this code here. My name is Legion, for we are many, hundreds of different software pieces which we can use. Two here, Plink and GCTA, we're going to work with today. Some of also often used Bolt LMM, Sage. This Regeni is becoming very, very popular. They allow to adjust for kinship and genetic relatedness, so that's why they're used a lot. This is just to mention, some pieces of software. And this is our kind of genetic model which you can run. So we have the phenotype and the SNP, age, sex, genetic principal components, and as a random factor, genetic relatedness matrix. If we want to, we can adjust for other covariates in our GWAS. So that's why we love using regression approaches, because we can do all sort of modeling in our analysis. Any questions? Yes?
So far, this suggests that we study BLA hypothesis.
Sorry?
So far, this suggests that we study BLA hypothesis. So how often is it that there are 3?
Yeah, yes. In most of the cases, we use SNPs, which are like biallelic SNPs. But in fact, even if SNP is triallelic, right, we can also analyze it as well, but just considering reference allele versus other allele, we just have more possibilities for this reference, yeah? Okay?
Why do we not include small indels?
We do include small indels.
Ah, okay.
Yeah. And new trend is to use CNVs, copy number variation. So I think it's becoming new trend. There was a couple of studies recently published for GWAS with use of copy number variation. So, but SNPs, because we use them because they are abundant, easy to kind of to genotype, and we know a lot about haplotypic structure.
But if we're talking about microsatellites, for example, they probably cannot be used for imputation.
We avoid them. We don't use them for GWAS. For linkage analysis, we do use STRs, but not for GWAS.
Also, in this model, epistatic interactions are not.
Epistatic interactions can be analyzed as well. The problem with epistatic interactions is the power, statistical power. It's quite, you really need very big samples. There are studies with with epistasis, analyzing the gene by gene interactions, and also gene environmental interactions in context of like GWAS. They are not that common, but they're there, so they're present. They can be done. This can be done, and this is being done. Yes. of course. Meta-analysis and replication, very important things. So meta-analysis can help you to boost your power. If your GWAS is not powerful enough, you can meta-analyze it with some additional sample and increase the power and actually find the signal. So meta and mega-analysis, meta-analysis, when we run analysis separately in different populations, then we provide an average matrix through inverse variance weighting or something like that. Mega-analysis, when we just combine several big populations into a single study and run this analysis. So there are some issues with heterogeneity of different populations in terms of like a phenotype definition, especially concerning clinical phenotypes in different countries. Clinical definitions and clinical diagnostics of different phenotypes, some phenotypes can be different. And in some cases we have clinically defined phenotype, in some cases we have self-reported phenotype. So heterogeneity might be a problem. And the big deal here is a phenotype harmonization as much as possible, so to make sure that we're analyzing more or less the same phenotype. There are large consortia dealing with some interesting, like most important phenotypes such as obesity or like BMI anthropometry. For example, GIANT consortium, they published a GWAS with 4.5 million individuals. I think this is a like world record in the sample size for a GWAS so far. So far, let's see how things develop. So let's say we have several studies, several GWAS run by 10 groups, and then we meta-analyze them and produce some pooled estimate of the relative risk, or its ratio, or effect size if it's a continuous trait. And replication, apparently we can be wrong. Right? We just may be analyzing some specifically weird population where there is this association, not necessarily it is true, just happen to be like that, or maybe it's specific to this population. So ideally, we need to replicate our finding in an independent population. So we just select SNPs which we consider statistically significantly associated and do the tests for these SNPs in a separate cohort, in a separate population. And if we still find statistically significant association, bingo, this likely the true association. Yes?
I have a question. Doesn't it may have medical importance if like one specific population has like some rare variants that influence the disease?
This might be the case. The thing is that we, in context of GWAS, we don't analyze rare variants. We focus on common variants. Yes, there are so-called accidental findings when we genotype somebody and find a mutation and it has clinical relevance. Then we can report it back to that individual or to the doctor, and then they deal with that. Yes, but yeah, apparently, yes, there can be things, for example, if we have a family in our cohort which share the same genetic variants related to this disease, and then it, sticks out as a signal of association. This may happen, of course. These findings, well, not so common, but everything is possible. So, but we consider common variants present in more or less in all populations, and so when under this consideration, we look at the additional cohort and seek for replication of our findings. Can be different, difficult, especially if we already generated a big data set, like big consortium. So where can we get another sample? This might be difficult. Sometimes we have a big consortium, we just split the data into discovery and replication. Okay, and post-GWAS analysis, as I mentioned, this is most interesting things. So first question, what are the associated loci? We found it here. Then next step would be what is the likely causal variant within this locus, right? We have many SNPs in this locus. Not necessarily all of them are actually making any influence on the phenotype. They are just simply in LD with a true causal variant, right? We need to identify which of these SNPs is likely the causal variant. Okay? This is called fine mapping. Then we try and make some mechanistic inference. What might be the actual functional effect of this SNP in terms of epigenomic regulation, of gene expression, for example, or the structure of the protein or something like that. And what gene actually this SNP belongs to, what gene it regulates, not necessarily is the one which is the closest to the SNP. Okay, as you know, SNPs, let's say in enhancer, may regulate a gene which is like 1 million base pairs somewhere else. Okay, so we need to find out which gene likely this SNP affects. Okay, and of course, if we have many genes identified, we can try and make some additional functional inference using analysis of pathways, genetic networks, and all that. So, one of the typical step is a SNP-based heritability. So I told you about narrow sense heritability. So SNP-based heritability is actually narrow sense heritability. It's heritability attributable to the SNPs which we analyze. So every single SNP contributes a little bit to the phenotypic variation. And then we summarize the impact of all these SNPs. This is how much heritability we managed to explain with our GWAS. Genetic correlations. If we're interested in shared genetic component between different phenotypes, we can actually calculate how much their genetic component correlates. This is what we are going to do during the workshops, SNP-based heritability and genetic correlations. Then top independent variants. So we have a locus with many SNPs. Many of them are significant. What we need to find, if there is one particular top independent SNP or there are several signals of association independent from another, one from another in this region. So this is what we also are going to do. Fine mapping, we are not going to do that because it's very time consuming, I'm afraid. So when we try and identify which is the likely causal variant in this region, colocalization, when, for example, we have several phenotypes and we want to see if these two phenotypes actually share the same genetic variant. Variant to gene mapping, so we identify variants and now we want to see what genes they belong to. Also not a trivial task, can be done automatically nowadays, but unfortunately it's time consuming. Pathway network analysis and fancy things like polygenic risk scores and Mendelian randomization. They are not part of the GWAS, but they use results of genome-wide association studies. Okay, I suggest to make a break. Okay, let's move on and talk about problems with GWAS. It may sound simple, but it's not that simple. So first thing is statistical power. One of the biggest problems until recently. I just had a conversation with one of you here who has a very small data set and cannot achieve statistical significance, which is expected for such a small sample size. What we need to achieve statistical power to get this p-value as a threshold for GWAS, which is, as I mentioned, 0.05 divided by 1 million tests. And 1 million comes from the approximate number of haplotypes. Individual studies are often underpowered. So that's why we need meta and mega analysis, which I mentioned before. Genotyping and imputation quality, those are important things which we need to take into consideration. Genotyping is usually less a problem because it's, these big companies, Affymetrix, Illumina, they're doing a great job in genotyping. Sometimes there are some issues when they cannot really properly identify the genotype, but it's a rare case. Imputation quality, I mentioned, you need to really deal with that. You need to set up a meaningful threshold to make sure that the dosages or genotypes you're working with are, more or less trustworthy. So to do all those things, we set up some filters when we run GWAS, which includes imputation quality and also includes let's say threshold for Hardy-Weinberg equilibrium, because Hardy-Weinberg equilibrium, if it's the p-value is very low, there is a huge deviation of Hardy-Weinberg equilibrium, it normally shows us the low quality of genotyping. And there are other filters which we can set. We will talk about this later. Confounders. confounders, they may affect the results, such as like population stratification. I'll give you an example later on. So we need to consider adjusting our model for some confounders, and also like consider things like a stratified analysis of people from this group or that group to avoid false positive, false negative findings. Phenotyping quality is a very important thing, and I will illustrate on a next slide. And replication is required. In some journals, they actually don't publish GWAS without replication. They just say, find another sample to replicate. So finding in a single cohort is not considered sometimes as a strong evidence. Things are shifting because we are working with very, very, very big sample data sets, like UK Biobank, 500,000 people. So why do you need to replicate it somewhere else? Apparently, you know, it's likely the proper finding.
I have a question.
Yes.
By replication, you mean just doing the same thing, but on another
in another cohort?
Or replication as finding like a biological reason?
No, replication in statistical sense, so replication in another cohort. And molecular analysis and all that, this is kind of thing aside. So what GWAS produces, list of SNPs, list of potential genes, and maybe some ideas about potential pathways and networks. The rest goes to the lab, so if people are interested, they can start working with mice, with cells, with, I don't know, God knows what, to find the actual kind of how this works. Because apparently here we're dealing with a statistical analysis of the data. It's good, but apparently statistics requires some kind of, in this case, some real molecular evidence. Just to illustrate the thing with the power, it depends on many things. It depends on the frequency of the risk allele in the population. It depends on the effect size of the risk allele. And here, some data from a Wellcome Trust Case Control Consortium. It was one of the first consortium to run like big GWASs at the time. How big you can see it's like, 5,000-10,000 people. Now it's considered very kind of small scale GWAS. So yes, so when we increase the frequency for risk allele and if we increase the effect size of risk allele, of course our power will go up. In this situation, when the risk allele is like 20% in the population and odds ratio is just 10%, 1.10, right? So this study, and this is the sample size, your study will be heavily underpowered. This is what your statistical power would be, 0.0002. So it's very unlikely you will find anything, okay? But so when you increase your sample, your power would increase. If allele has reasonably strong effect, then your power will be higher. And if your allele is more or less common in the population, also your power would be higher. So you can do all these calculations before you run the analysis to make like some assumptions of how big a sample you need. Or you can do this, of course, after, when you didn't achieve statistically significant results. You want to calculate how much power you have and then plan your subsequent study. So there are calculators online where you can go and actually make some assumptions about effect size, case control ratios, total sample size and all that, and see how much power actually you expect. Population stratification, just want to illustrate it with a very funny example. Beware the chopsticks gene. So the people who run this analysis, they were interested whether or not there are genetic factors underlying your ability to use chopsticks. I cannot use chopsticks. Many people try to teach me. I simply cannot. My fingers don't work. I don't know how to do that. So, and possibly there are many people like that, and they were interested whether or not there is a genetic factors contributing to your abilities to be able to, your ability to use chopsticks. They ran genetic analysis, compared people who can or cannot use chopsticks, and found massive, very, very significant association, right? And when they start, and they call this gene sushi, stressful use of selected hand instruments gene. Yes, very funny, very romantic. But then they started, and it was in the HLA region, so which is responsible for immune response and all that. So how on earth can this be associated with your ability to use chopsticks? But when people started digging into the sample, they realized that actually they effectively compare Europeans who normally culturally don't use chopsticks and Asians who normally culturally use chopsticks. So they simply compare two very different, genetically different populations, which, and for the phenotype, which is culturally driven rather than genetically driven. So it's a simple culture. It has nothing to do with genetics. So if we have the situation like that in our population, when we have a mixture of populations with different frequencies of some alleles and different frequencies of the phenotype or different manifestation of phenotype of interest, simply not because of genetics, but simply for cultural or social, whatever other factors. Okay, this phenomenon is called population stratification, and it leads to false positives like this case. Okay, so this is important things. How we adjust for that? We can adjust for that. First of all, we make sure that our population is ethnically or genetic, from the point of genetic ancestry, is more or less homogeneous, belongs to kind of, let's say, European, Asian, African population. Second, in our model, we adjust. We add genetic principal components, which can eliminate some kind of subtle effect of population stratification. And more advanced method is to use genetic kinship matrix, which does the further adjustment for relatedness, hidden relatedness, and population stratification between the individuals. Phenotyping quality, which I mentioned, here is the interesting example. So in this study, they looked at nuclear cataract as a phenotype, and it was measured using two methods. One is called Wisconsin cataract gradient system. It's very highly accurate method, very correct measurement of a cataract. And the lens opacities classification system 3. It's good, but not as good. It measures with errors. So it's not a very good method to test for a cataract. And when they run GWAS using Wisconsin system and this LOX3 system, SNPs were different. So it's a kind of the same phenotype, but measured in different ways, produces different results. So it's not necessarily the same phenotype then, right? So you cannot really use it for meta-analysis. You cannot use this one phenotype and then another for replication. Okay, so phenotyping quality is an important thing to consider in context of GWAS. Replication also, I mentioned to confirm true association and deprecate false association. Of course, your replication, first of all, you have to have a big discovery sample size. Then you also have to have a big replication sample size, okay, for the statistical power. Ideally, you should have an independent data set. Practically, this may be difficult, as I mentioned. So you already have a very big consortium with a very large sample size collected everywhere in the world. There are no other data set in the world with the same phenotype. Where exactly are you going to take the sample for replication? That might be difficult. Normal practice now is we make a consortium, we collect as many samples as we can, then we split them into discovery and replication. This is a good way. Less good way, worse way, when we have a sample, a single sample, which we split into discovery and replication. For example, we have like UK Biobank, 500,000 individuals. That's a lot. For GWAS, more than enough, very powerful. We'd say, okay, if 75% would be our discovery, 25 replication. It's not that bad, but not ideal, because if you have kind of something wrong in the whole population originally, right? So your error will persist in your replication sample. Still, it's one of the ways to handle a replication problem when we don't have an independent data set. Of course, the same or at least very similar phenotype must be there. And genetically similar population can be studied. So if you made your findings in Europeans, so you better try and replicate it on Europeans first. Of course, you can look at other ethnic groups, but if you don't replicate, it not necessarily means your signal is wrong. It just may be in this ethnic group. There is no such association for whatever reasons. Okay? Very important thing. If your association is true, your effect size is expected to persist in like any sample. Okay, your p-value may be different, of course, because of the sample size, but if it's the true signal, and that's the impact of this genetic variation on the phenotype, let's say 30%. And if it's true association, you should expect in your replication sample, the effect would be more or less the same. If it's not, then kind of you should consider your replication with caution. There are some also very weird situations which happened to me when I run the analysis in the discovery sample and the replication sample for a SNP. There was the significant association, but the effect size was reverse. So in discovery, it was positive association of this allele with the phenotype. In replication, it was negative association. Can we consider this as a replication? We cannot. So something weird is going on. All right. Yeah, any questions? Yes?
What if someone doesn't have two independent populations to study over? Can you, or is it, I don't know, a good solution to do something like a K-fold cross-validation?
Well, yes, you can. Well, you can simply try and publish whatever you found without replication. I mean, for some people actually argue that if you analyze a very, very big sample size, very big phenotype, very big data set, you don't even need a replication because, you know, it's more or less trustworthy. Some people disagree, but, you know, then you can use a triangulation of evidence. So apart beyond from your findings for this SNP with for this, if you also have some molecular data, if you have some data from UQTL, or if you have some additional data, you can try and convince reviewers, people that this is the genuine finding and this is true. It really depends on the journal policies and really depends on kind of editorial policy or reviewer kind of attitude towards replication. So, but there is this trend kind of not necessarily you need to replicate anything if you analyze like 5 million people, so apparently it's all correct, more or less. Yes? More questions? No? Okay. So, there are several catalogs which summarize results of GWASs ever done, being done, and you can get pretty much all this data for various phenotypes done in different populations. There are several catalogs, so one of them is called GWAS catalog. You go there, European Bioinformatics Institution, you type, I don't know, cataract, and it gives you the list of published GWAS results. And in many cases, it gives you the link to summary statistics. So the actual results from GWAS, which you can grab and use for your analysis. This is very handy. Another requirement nowadays is if you run a GWAS, you need to deposit your summary statistics somewhere, so it must become publicly available. So there's this GWAS catalog, and now for example, there are several large programs, genetic programs, includes UK Biobank, so people from the UK, 500,000 people, and several groups have run GWAS for every single phenotype which is present in this cohort, and all this data are deposited. You don't need to run your GWAS, really. You can go there, grab the data, and do whatever you want. The problem here is that, of course, they made their assumptions, they made their adjustments, not necessarily what you want to consider in your study, so you treat them with caution. But if you want to have a quick look at what's already known about this phenotype in terms of genetics, you can just go there and grab the data. Another big data set is FinGen, so it's an analysis of 450,000 of Finnish people, also for very many clinical endpoints for diseases. Another recently released very cool data set, Million Veterans Program from the USA. They analyzed kind of 1 million veterans and for many clinical and non-clinical phenotypes. And then they also run a very good analysis on Europeans, on people from Latin America, not with Latin American ancestry, African Americans, and some Mexicans, and some other subpopulations which are present in this program. And they also carried out trans-ethnic meta-analysis. So it's very, very important, very interesting data set. So you can grab them all, analyze them, and see whether or not you can, bring them to the lab and maybe make some discovery on a particular gene. So if you want to know more about GWAS, there are several papers which actually describe the concept and also describe our success in running GWAS, what we have achieved in like 10 years running the GWASs, 15 years, and so on. So these are some useful references for you. So you can feel free to read and make your own opinion about the usefulness and whatever else for GWAS. Okay, so that was the introductory part for GWAS. And unless you have questions, I will move on to the second part and talk about some useful applications beyond the discovery of genetic variants associated with diseases, namely about polygenic risk scores and Mendelian randomization. So, polygenic risk score, it's one of potentially practically important outcome from GWAS beyond the discovery of genetic factors. It's based on so-called polygenic or infinitesimal model that have been proposed by Ronald Fisher in 1930s, I guess, or 20s even. So the complex trait is controlled by an infinite number of loci, each with an infinitely small effect. So we have many, many genetic variants in our genome, SNPs, for example. Each of the SNPs contribute a little bit, teeny tiny effect into a phenotypic expression. When we summarize them, the effect becomes quite big, pronounced. So, and this is the idea behind polygenic risk score, which is a weighted or sometimes unweighted sum of a large number of risk alleles. So let's say we have several individuals with several SNPs, and for this SNP we have association measurement of the effect size from our GWAS, odds ratio or regression beta, and we simply multiply the number of genotypes in this individual genome for this SNP by the effect size. For this SNP, this person has 0 risk alleles, so 0 times 0.1, which is the effect size for this risk allele. For this SNP, it has, he or she is heterozygous, so it has 1 effect allele times effect allele sample size of this effect allele. Sorry, effect size for this allele. And so on. We do this for many SNPs, and we calculate the sum of results of this multiplication. So when we have a quantitative phenotype, 0.2, 0.5, 0.8. This is just the example. So this is how it is calculated. This is a simplified version. Of course, there are more sophisticated ways to calculate it, but for simplicity, it works like that. So now, when we have many people for which we identify polygenic risk score, we can build a distribution and consider some thresholds for high or low risk of the disease of interest based on this polygenic risk score. So this right part of the distribution would be like high risk individuals. This part of the distribution will be low risk. They have low polygenic risk score values, and in the middle there will be some kind of intermediate risk for this clinical phenotype.
Sorry.
Yes.
Can we assign different weights to different alleles in this model?
For a single SNP, the effect allele will be the same.
Okay.
For this SNP, so across all individuals, the effect, each SNP will have its own effect size. Each SNP will have its own effect size. Okay? So for different SNPs, the effect sizes would be different, of course. So for this SNP, the effect size is 0.1. For this SNP, effect size 0.2. For this SNP, 0.3. Okay, then we consider this individual who might be homozygote for non-effect allele, heterozygote and homozygote for non-effect allele. Accordingly, we multiply by this effect size attributable to this particular SNP. Yep. Okay, just to, there's tons of literature about polygenic risk score and attempts to bring them into clinical practice. Not very successful so far because in many cases the predictive ability of polygenic risk scores are not that good to bring them to clinical practice. With a few, there are some exceptions, and people are still optimistic about that. So potentially at some point we may consider bringing polygenic risk scores to clinical practice to make some predictions. This is one of the studies which kind of shows the potential of the use of polygenic risk score to change the healthcare system in the UK. So it's a polygenic risk score for the risk of breast cancer based on common variants or predisposition to breast cancer. It's not about mutations, not about driver mutations, it's predisposition to breast cancer. So in the UK, mammogram screening is initiated for women at age of 47 years old. Because from population data we know that at this age there is a 10-year risk of breast cancer in average woman. So when a woman turns 47, within 10 years there is a high risk for this woman to get breast cancer. So, and that's why the healthcare policy recommends women to go to mammography at this age. When we can see the polygenic risk score for breast cancer, so they are profiled here, so this is the risk, population risk of breast cancer, right? So these are people from 99 percentile of the distribution of polygenic risk score, and they reach this threshold at age around 30 years old for the risk of breast cancer. These women, 95, 99 percentile of the polygenic risk score for breast cancer, about 32 years old, the risk goes beyond the population risk threshold, and so on. People from left side of the distribution, low risk, less than 1%, they actually never achieve the clinically relevant threshold for the risk of breast cancer. So now, if this is all true, right, and if we bring this to clinical practice, we may say, so women who have this high risk should go to mammography at 30 years old. Don't wait until they turn 47, because it will be too late. They already are well beyond the population, or the kind of the expected risk for breast cancer. Those who actually are very low threshold maybe don't need to go to mammography at all, because their risk is very, very low. Or at least they can be, for them, something else can be recommended rather than mammography, which is like a costly business. Okay, so this is one of the examples. It's not in clinical practice. This is so far still theoretical considerations, but this is how potentially this might be working. It's a polygenic risk score. So how this is done? You run a GWAS in a large discovery sample, the bigger the better. Then you make a list of SNPs with various significance thresholds. So you start from most significant ones, then you go down. So you take these three first, then you take these 1, 2, 3, 4, 7, 10, 11 above this threshold, then you take all of those, then you take all of those, and so on, and go down until, I don't know, SNPs with no significance at all. Okay, so, and then for every set of SNPs, we calculate polygenic risk score in a target population and see which of these list of SNPs provides you with the the highest explain variance, okay, the highest effect size for the phenotype of interest. Right? Then we say this is our polygenic risk score. Let's say this subset of SNPs gives us the highest heritability estimates or coefficient of determination in a regression model. Then we build a distribution for this polygenic risk score and start playing with the thresholds to see if we can make meaningful prediction for phenotype of interest. So we compare this with some gold standard. So we classify individuals into high risk, low risk, and compare them with some like gold standard and see whether or not polygenic risk score predicts anything meaningfully. Okay, so this is approximately how it works. So I would like to illustrate with my own study a potential practical implication of GWAS using polygenic risk score. I was interested in chronic back pain phenotype. Chronic back pain, it's the #1 cause of the disability worldwide, #1 cause of job absenteeism worldwide, costs a lot of money to healthcare, to the society, and there is a reasonably strong genetic component into your risk for chronic back pain. So the problem is treatment of chronic back pain, in particular stratification people into meaningful groups for the developing chronic back pain, or kind of if you have an acute episode, not necessarily developing it to a chronic back pain. The thing is that in some cases, for people who experience like serious acute episode of back pain, there is a certified kind of recommendation for clinical management of these people, and some people simple kind of the use of painkillers for some period of time will work very well. For some people would require painkillers plus physiotherapy, and some people on top of that would require psychological support. Okay, so that's three kind of clinical ways for people with a serious episode of back pain. There are some clinically kind of relevant existing instruments to do this certification, but they're not. List them, some of them here, like START-BAC screening tool, allocates you into 3 treatment groups. Örebro musculoskeletal pain screening questionnaire, prognosis tool to determine the risk of long-term absenteeism from work. Okay, there are some clinical tools, but they're not working that well. So if you look at for them to predict risk of chronic pain, the area under curve, so which shows the kind of the potential, the predictive potential of your stratification tool, 0.55, just slightly bigger than random guess, like in 50% you may guess yes or no. This will become chronic. In 50% case, you will be correct, right? If you just make a random guess. With this tool, it's 0.55, slightly better. In Örebro screening tool, it's not that bad, 0.67, 69, sorry. So it's better, but not perfect, because for clinical applications, something which is lower than the 0.7 kind of is not considered seriously. Okay, so the question is whether or not we can help improve these diagnostic tools with use of genetic factors, right, which predispose you to high risk of chronic back pain. So I have done this analysis. I used the UK Biobank as my discovery sample to identify SNPs for polygenic risk score and made prediction for back pain using Twins UK. I ran different thresholds, and I tried to predict disabling low back pain. It's when you cannot like put on a sock, when you can, sit down, then stand up from a chair, when you can, stand from the bed and all that. So disabling low back pain, you cannot move. And any low back pain, not necessarily disabling. So I calculated this polygenic risk score, identified the best predictive kind of set of SNPs, and then this is the prevalence of low back pain in different percentiles of the distribution of polygenic risk score. If we see up to like here, like 0.1, people here have very low risk of low back pain. And after like 90% of the percentile of this distribution, we have very high risk of low back pain, and everything in between of them is actually somewhere around average risk in the population. This red line is like an average risk for back pain in the population. And then I compared, so I calculated those ratios for the risks of back pain from the left and right side of the distribution based on these percentiles, 0.2, 0.8, 0.1, 0.9, 0.05, 0.95. So I compared the low and the high risk against the intermediate risk. And actually we see here, for example, statistically significantly lower risk for people from this percentile, below this percentile of polygenic risk score compared to intermediate risk. And in the high risk group we have 1.46, so seems to be there is a good potential to predict the higher or lower risk of back pain using this polygenic risk score. And actually you can look at these bar plots for disabling and for any low back pain. So people from high risk group, that's the risk of disabling low back pain, intermediate and low risk group. So here's very nice step. Like ladder-like distribution. So, which makes me able to make the statement polygenic risk score for back pain is promising. Whether or not it's clinically relevant, we don't know yet. What we are doing now, we are trying to run the analysis using together these clinical predictive tools and polygenic risk scores in a single model to see whether or not the use of polygenic risk scores on top of clinical tools would help make better predictions.
I wonder how you consider the age in which it starts?
In this model we don't. In this model we don't, but it's like important thing to consider, of course, yes. Yeah. In this model, the thing is here, they are all, in UK Biobank, they are from 40 years old and above. In twins here, they're also like 40 or something and above. So they're already kind of beyond the youth, right? So they're more or less on the same age scale. Any more questions? Yes, I forgot to show you this. It's a very, yes, it is promising, but area under curve is 0.56. Not very good. And this is the distribution of a polygenic risk score in controls and cases. So you see in cases this shift is towards this side, but you know, still it doesn't mean that it's useless. If we look at some percentiles, so people from the 99th percentile had almost double the risk of chronic back pain. So which is actually good. But as I said, whether or not it's clinically relevant, we don't know yet. Mendelian randomization. Have you heard of it? No? Yes, Diana, heard of it? Use it? No? No? Okay, I'll teach you. Yes, Mendelian randomization. So what it does and how it works. Let's say we identify an association between some kind of a biomarker and a disease. It's a statistical association. Apparently, it doesn't tell us about whether this biomarker actually causes this disease, right? So it's correlation versus causation, right? It's a central question in epidemiology, because what epidemiology is looking at is like, you know, finding relationships between some modifiable exposures and some outcomes of interest, and then testing whether or not these associations are causal or not. If they're causal, then we can try and modify our exposure and actually prevent the disease onset. Right? It's potential situation why this association are present. First is causation, right? So when we have a biomarker of like, I don't know, the high blood pressure, for example, right? Whether or not it's the cause of cardiovascular events, right? Causation, this is what we want to show. Maybe it's just the effect of some common confounders. So both, let's look at the confounder which influence both the exposure and the outcome, artificially creating relationships between exposure and outcome. Another situation is a reverse causation, when actually it's not the biomarker which causes the disease, right? It's the disease which causes the change in the biomarker, right? So Mendelian randomization actually can help us to test if this scenario is likely. So that's one of the definitions. So Mendelian randomization is a form of instrumental variable analysis, a common approach in causal inference that uses genetic variable to test whether an intermediate phenotype causes another phenotype. It is done by the analogy with randomized controlled trials. So this is the standard way in basic epidemiology to identify causal effects of some exposures. So we take a big sample, homogeneous for some phenotype. Then we randomize it into two groups, just randomly allocate them into like treatment group and placebo group. Then we exercise some like intervention. Let's say we give to one group some experimental drug, and another group we don't give anything or give placebo, right? Because these groups are randomized, they are like equal in all parameters. If we see an association of this intervention in our intervention group, so the only cause for this association would be the causal effect of this intervention, right? Because otherwise they're completely like equal. There's nothing else which may influence it. So this is how this is done in clinical epidemiology and basic epidemiology. Mendelian randomization actually uses the same kind of idea, but randomization is not done by epidemiologists. It is done by the Mother Nature, right? So based on random segregation of alleles in the population, we actually randomized in terms of having different alleles associated with the phenotype of interest, right? So, and if we have an allele present, strongly associated with the phenotype of interest, so people from this group would have higher risk of this disease compared to control group which has other allele. Okay, so this is randomization which is done by the nature, and confounders are equal between groups. We just take the whole population, they all live here, they eat the same food and all that, so they are more or less equal. Then we compare outcomes between group from this exposure group, which is defined by the presence of allele of interest or several alleles of interest, versus another group which is defined by the absence of risk alleles. And if we see the association, then we assume there is a causation between this exposure of interest, which is like a, the same thing like blood pressure, for example, right? And the outcome. So this is the idea behind the Mendelian randomization.
What would you try that we try to split our sample based on wild allele and keep randomness for other alleles?
All alleles, everything is random. But in people who have a high risk of disease through the effect of exposure, there will be a bigger combination of alleles predisposing to like high or low level of this exposure factor. They're all randomly distributed in the population. But if they are associated with the phenotype, they will come together. Yes.
In this first case, say we actually rather mean that we're separating groups not by alleles but rather by haplotypes.
We're not separating groups as such. Yeah.
Yeah, but I mean, well, okay, it's the exposure and the control.
The exposure group will have a greater number of alleles associated with, let's say, high level of a particular like exposure phenotype.
But we will probably not be able to identify the exact alleles.
No, we know them. We know all these alleles. We know these SNPs. We know the SNPs.
But if they come as a haplotype,
Yeah.
then we don't know which one exactly. It doesn't matter.
It does. No, the thing is that we don't even need a haplotype. We remove the haplotype. We only keep a single SNP from the haplotype with the strongest effect.
Okay.
Yeah, this is to avoid duplicating signals, because every SNP in the haplotypic block will be having some little impact on the phenotype. We don't need that. So the same signal done several times again and again. So we don't need that. We remove it. So we do clumping, and we leave a single SNP from this haplotypic rock strongly associated with the phenotype and keep it in our model. Okay, so these are some assumptions which we make when we run Mendelian randomization. It's not that simple thing. So we use genetic variants from a very large GWAS strongly associated with the phenotype. We remove some SNPs in LD. This is what you asked about. So we remove some SNPs in LD, and we test how strongly they are associated with the phenotype. The first assumption here is that genetic instruments must be very strongly associated with the exposure. Okay, so this is the relevance of our instruments. Second assumption, the genetic variants must not be associated with confounders, okay, to avoid bringing false positive signals. And the genetic instruments must not be associated with the outcome by any way rather than through this exposure. And these are this is taken from the real paper. So some actually specific ways of handling things, how many cases they have, controls, some tests which they carried out, and some parameters for choosing the SNPs. This is just to illustrate whether or not type 1 diabetes causes polycystic ovarian syndrome, right? So this is the Mendelian randomization study asking this question. One word of caution. There are, as an editor of a journal, I receive hundreds of papers just randomly, analyzing potential causality for some randomly selected phenotypes. Okay, this is wrong. The first thing you have to do is to make a reasonable assumption about causality between these phenotypes and then phenotype based on either molecular evidence or based on epidemiological evidence. All right? This is the thing. If you want to run a Mendelian randomization and want to get it published, you have to make sure that you have a valid hypothesis. So this is the trend in publications on Mendelian randomization. Technically, it is reasonably simple, same as GWAS. There are software packages which actually allows you to run Mendelian randomization for any combination of phenotypes with a few clicks of the mouse. And people do that and send off papers for publications which are absolutely meaningless or incorrectly implemented, done with errors and all that. But the trend is here just because it is available. Just to give an example, I received a paper when they analyzed causal relationships between Alzheimer's disease and myopia. Alzheimer's disease onset is, 60 years old. And the onset of myopia is 2, 3, 4, 5 years old. Alzheimer's disease cannot be the cause of myopia, because myopia starts many, many, many years before Alzheimer's disease. And people just don't, I don't want to say rude words, but, you know. There's this guy, George Davis Smith. He kind of one of the inventors of this method, and he stopped inventing this method. He's starting like wandering around the globe, telling people be very careful with Mendelian randomization, okay, to avoid these, you know, crap papers and kind of meaningless papers. And this is the paper which I recommend you to read. And also there are some video on YouTube when we do the project. If you choose to do the project with me, I'll give you the links to these videos. If you don't, I won't give you the links. Okay, let me give you an illustration from my own work. So I don't give you too many details. I just give you like this kind of this snapshot of the idea with what can be achieved with use of results of GWAS. So one of them is Mendelian randomization. So back pain, again, so there are many factors which are associated with back pain and considered as risk factors of back pain. Like female sex, older age, low social economic status, obesity, degeneration of spine disc, and many other things. The question is, if these are associations, happened because they happened, or are those things causal factors for back pain? So we run a Mendelian randomization to test for some risk factors, years of schooling, never smoking, alcohol intake, and overall activity, and then some other things. And we've done it in two directions, so forward Mendelian randomization to test if these are the potential causal factor for back pain, and the other way around to see if actually chronic back pain is the cause of at least some of those associated factors. So this is the finding. Fewer years of schooling, smoking, greater alcohol intake, major depressive disorders are causally associated with low back pain. So those are the factors which, if avoided, may reduce your risk. Those are modifiable factors, right? We can change them. May reduce the risk of back pain because they're actually the cause of back pain. And in reverse, back pain is actually also the cause for smoking, the high risk of smoking, and for greater alcohol intake frequency. Right? So if you experience back pain, so smoking, alcohol, they actually can help you kind of live with back pain a bit easier, so which makes it the causal factor for you to restart smoking or start smoking and drink more alcohol. And the interesting thing about smoking and back pain, so how can smoking be the cause of back pain? But in fact, there are very interesting studies showing that actually the composition of intervertebral discs and even their color is different in people who smoke and who do not. So potentially smoking compounds, like thousands of different harmful compounds, right, from the tobacco smoke, they somehow reach intervertebral disc and somehow lead to the high risk of back pain. Any questions? So if you smoke, you better stop.
Why is there no exercise mentioned in this?
Sorry?
Why is exercise not mentioned?
Because we didn't find it statistically significant.
It seems weird to me that it's not statistically significant. Because
this is science. Yes, there are many things which seem weird, but no. The thing is that first of all, this might be a issue with the power, right? Second, this might be. The thing is that, for example, physical exercise is done for leisure. You go to gym, you go for a walk, you go for jogging and all that. Actually reduce the risk of pain. And physical activity associated with your job increases the risk of back pain. All right? Here we just considered all sorts of physical activity, and maybe they just kind of nullified each other, but potentially, I don't know for sure.
Maybe it's just classified differently. Maybe the pain which professional athletes have, maybe it's not classified as chronic.
The thing is that we just take the people's word for it, whether or not you have a back pain lasting for at least 3 months. The causes of that, why do you experience it? Because of your job, because of your trauma, or because of what? We don't know. Okay, we just. It's a broad. It's not a diagnosis even, right? It's not a clinical diagnosis. It's just the self-reported complaint from people.
So if someone injured their back but were falling on it, it also would be.
Yeah, if they injured their back and the pain lasted for 3+ months, then they will be there as our cases. Yeah.
Actually give like a clinician statistics from like clinicians or hospitals.
Yeah.
The power is less.
In Europe, like 40% of people would experience at least one episode of severe back pain after age 30 or 40, I can't remember for sure. It's very common, and also I mentioned the cause number one of job absenteeism and disability worldwide. Millions of people have kind of experienced back pain serious enough to kind of to stay home or to feel disabled for quite a long time. It's a massive problem. In UK Biobank, the prevalence of chronic back pain, I think it's about 45%. 40 plus, can't remember exactly.
Isn't it a possibility that when we find a causal relationship between these factors, that these are only causal because they are actually confounders, but we cannot measure or we don't measure the things which are really causal?
Yes, this might be a problem, yes. We assume that because of the way how Mendelian randomization works, the effect of confounders is minimized, but we cannot get rid of them completely, of course. Right? They still may be present, and there are some kind of statistical tricks how to try and minimize that. It concerns the sample size, it concerns the number of SNPs, it concerns the effect size for the SNPs, and so on. And plus, there are several, usually you don't rely on single method for the analysis, you rely on different methods which make different assumptions, statistical assumptions. If they all kind of concordant, right, then likely, it's working well. In terms of confounders, there are several ways how to handle that, apart from what I mentioned, and one of them is use multivariable Mendelian randomization, when we actually include confounders into the model to see whether or not our causal association with the exposure, persists, even if we put we randomize, additionally people randomize for this confounding factor. Any more questions? Okay, that's the project I want to offer you. Sexual behavior as the cause of chronic disease, NMR study. Yes, I decided to make it interesting. Okay, if you want to know more, do join my project. I will tell you if you join my project. No, in fact, no. Okay, so there are findings that show that, including my own work, that age at first sexual intercourse actually is a predictor of chronic disease in future life. Age when you have your first child, 2, it persists for males and females, so it's not sex specific. Right, so it's somehow related to. Apparently, you cannot. I mean, there is no way how the age when you start your sexual life would affect something. Apparently, it's like a proxy phenotype for something else. I have 2 assumptions. First is your behavior. If you like a risk-taking behavior, we definitely have a genetic component, which includes some crazy things, even kind of taking the risk to start sexual life when you are, 12 years old or something, right? And another thing is anthropometric factors. So because to start sexual life, you have to you have puberty, you have to be mature enough to actually to have it physically. So maybe people who start sexual life early, they actually maybe growing up and mature early than other, they developing quicker. And because they're developing quicker, they may be aging quicker, and this brings over diseases. So these are my two kind of hypotheses how this might be related.
Oh, may I ask another question? So all the cases from the study are like consensual intercourses. Let's put it like that. So
No, not all, no. The study which so in UK Biobank, right? This is what I used. Apparently, it's 500,000 people, or maybe in some cases it was unconsensual intercourse, but I'm not sure there is this data to. I actually no. It's a good question. I need to double check and possibly exclude those. But
yeah, because I think you can find some interesting.
No, I mean definitely. I mean apparently there are not that many to kind of significantly influence any associations. Yeah, and there are studies from other cohorts as well, or more or less well characterized, showing similar things. Yeah, so I think this is true. The question is why is that? Yes.
There are some cultural subpopulations where this starts early.
Yes.
Like
Yeah, no.
some
Yeah.
Yes. And typically there is no it's not about consent, it's just tradition.
Yeah.
And people typically don't even need to be mature enough.
Yeah. I know.
Those that start the marriage. Will it be possible to include them actually to distinguish, because this cohort will help to distinguish early maturation, early speeded up maturation, speeded up life, from those who just have inappropriate behavior, because they don't have inappropriate behavior.
Yeah. It is difficult, yes, apparently. So this is that's why I've done it in Europeans only, which are less likely to be Muslims, for example, right, than non-Europeans. But the factor to take into account, I don't have means to do that in UK Biobank because there is nothing about that in UK Biobank. But potentially, yeah, of course, this is interesting. I mean, currently there are lots of social factors, heavily kind of compromised by social factors, including like faith and traditions and all that. It's not a simple thing. Right, yeah, totally agree. And when I mentioned this to doctors, the first thing kind of was, yeah, but this is like social factors, so why do you bother? So, who knows? Maybe yes, maybe not. Right, let's move on. My project, everybody's welcome. And the workshop. And I think we should take a break.